Well, you know, since it is a preclinical product, that's exactly what I'm going to talk about. So since we're the most recently public company, I think we get the honor of having the longest forward-looking statement. To summarize, we're developing a product that is for the central nervous system. It's a glial cell product addressing a huge unmet medical need, multi-billion-dollar opportunity. As you heard, initially we're looking at ALS, but after the ALS starts, we intend to look at other diseases such as multiple sclerosis, spinal cord injury, and down the road, stroke, Parkinson's, and Alzheimer's. The product has a mechanistic approach that really lends itself to a wide range of neurodegenerative diseases. Very strong patent position. As of last October, we're a public company. We did a reverse merger into a clean shell. Teva, previously Cephalon, was our lead investor, and we're now targeting IND end of 2012, which is this year, and clinical trials next year in ALS with Johns Hopkins, Nicholas Maragakis as our PI. A lot of management experience at the company. The one advisor I'll point out is Mahendra Rao, who is the scientific founder, was recently appointed director of the new Center for Regenerative Medicine at NIH. So what we're looking at is how do we improve the lives of these millions of patients that are suffering from these diseases that there are really no treatments for. As mentioned, our initial target, ALS, multiple sclerosis, spinal cord injury, and future targets will be in the brain. The opportunity is huge. There are no effective treatments. Patients are desperate. Our solution is to use what we've trademarked as Q cells. These are human neural progenitor cells. I make the point that they are not a stem cell. They are further down the lineage than a stem cell, which means that they have the advantage that they only produce certain cell types. We don't have to worry about removing undifferentiated cells that might potentially form tumors. We're targeting the cells that are the natural support cells of the central nervous system, the brain and spinal cord, with proven support mechanisms. I like to use the analogy that these cells are mini factories. You've heard that before in today's presentation, but what they're doing is not producing just one growth factor, not just producing one mechanism such as remyelination. They're producing a full complement of growth factors. They're removing toxins, they're providing nutrients, and they're remyelinating. So it's really all the support that you need for neurons in the central nervous system. Strong patent position. We have 14 issued, many more pending. We have patent protection. Am I making that noise?
I think just you're close to the...
I see.
Yeah.
Patent protection out to at least 2030. So what do these Q cells do? What they do is they repair the neurons in the central nervous system. The neurons, shown on the left, are analogous to the transmission lines. That's how the electrical signals are connected. They're the guts of the CNS, but in fact, they're the minority of the cells, and they cannot function without the cells shown on the right, the glial cells, which produce all the factors and myelin and other support needed for these neurons to function normally. And so the target that we're looking at is to use these Q cells to repair the neurons rather than trying to replace the neurons. This slide is a little complicated, but I think it illustrates an important point and really highlights some of the differences between what we're doing and companies such as Neuralstem or Reneuron that you heard about are doing. If we look at what's shown in that little pink blob, the neural stem cell, that is the true stem cell of the central nervous system. It forms neurons as well as the support cells, the glia shown in the blue. We have taken a cell type that breaks off after the neural stem cells, so we are not getting the neurons. And we view that as a good thing. The neurons are viewed as an unwanted cellular phenotype that could potentially cause toxicities, whereas we are only producing the glia, which give us the two support mechanisms, the oligodendrocytes and the astrocytes. And in fact, if we look in an animal model, that's exactly what we get. Everything shown in green is myelin produced by implanted human cells in an animal model. Everything shown in red are human-specific astrocytes. We've looked extensively, and there are no neurons produced by the human cells. We have developed a manufacturing process that is very robust, very reproducible, with defined release criteria. And this is an allogeneic product, I should mention. So look at some of the animal efficacy. Here, if we look at myelination, what we see here, this is a mouse brain, a shiver or rag-2 mouse that has a defect in myelin production. A single treatment, a year later, we see long-term survival of these mice, and everything shown in green is myelin production, extensive myelination, and what you can see, not quite so well in red, but those are stains for a human-specific nuclei. Q with its collaborators at Johns Hopkins have done similar studies and actually have gotten 80% long-term survival in our first study. So that's extremely exciting because all the control animals die at about 120, 130 days. And I should mention this is a much more extreme disease target than what we're looking at in the human, because here the entire central nervous system is lacking myelin. Looking at spinal cord injury, this is collaboration with Itzhak Fisher, Drexel University. We see amelioration of several effects, cyst formation, glial scar formation. That's a very important factor because one of the reasons that spinal cord injuries don't heal is that you get a lot of scarring and the cells that would like to heal them don't have contact to, the sites that need healing. And then I don't show it in this slide, but it's shown in the publication. We also had a beneficial effect on bladder dysfunction, which is an extremely important finding for spinal cord injury. When we go to Lou Gehrig's disease, huge unmet need. A lot of people don't appreciate it, but the incidence of Lou Gehrig's is comparable to the incidence of MS. It's just patients don't live very long, so the prevalence is much less. 1 in 800 deaths is due to ALS. Huge unmet medical need. What is known in ALS is that even though the motor neurons are dying, and that's what causes the symptoms, it is the non-neuronal cells, and in fact the support cells, the astrocytes that are diseased, that are actually killing motor neurons. So our goal is to replace these diseased astrocytes with normal, healthy astrocytes as a method of preserving function of the neurons. And what we've shown with our collaborator Nick Maragakis at Johns Hopkins, and this was published in Nature Neuroscience, is that in the rat model of ALS, using the rat homolog of our cells, we get a nice increase in survival from the time the disease onset occurs, which is when we implanted the cells. We have almost a doubling of survival. And these cells were implanted cervically in the neck area. The target is ultimately to prevent respiratory loss because the neurons that innervate the diaphragm die. And we showed that we could get a sparing of that and also a sparing of forelimb function. So those are very significant. These findings led to a $4 million NIH grant to help fund the preclinical development. So some of the advantages of our product, and I list these here not only for other cell therapy products in this area, but even small molecules looking at central nervous system diseases. One, from safety, it's a normal cell in its native environment. We don't get the neurons, which I mentioned were an unwanted phenotype. Very importantly, it's a local treatment. The cells don't leave the CNS, so you don't have systemic toxicities to be worried about. From an efficacy side, we get both neuroprotection as well as remyelination because we produce both types of support cells. Widespread distribution in the central nervous system means more cells can go at the target site. These cells migrate in the central nervous system, and multiple mechanisms allows us to target a wide range of diseases. From the manufacturing side, it's a cost-effective manufacturing. We don't have to differentiate the cells into a different cell type, and again, a very strong patent position. So our clinical trial plans are to start off in ALS, a phase 1/2A trial. We'll be in patients. We'll start out local injection into the spinal cord, dose escalation. I can't read that. Oh. It's a problem with bifocals.
You're getting near the end of your presentation.
Yes, I will be within that time frame. Safety measures obviously are the primary endpoint, but we will look at measures of efficacy. We will be looking at decline in respiratory function. If there's any leveling of that, that's obviously exciting. We'll also be looking at electroimpedance myography, which is a very nice measure because it's a very quantitative measure of muscle function. And then obviously go into the phase 2-3. As I mentioned earlier in the study, we believe that there's a nice benefit of these cells for demyelinating diseases and would like to go into clinical trials in demyelinating diseases, starting off with multiple sclerosis, transverse myelitis. Transverse myelitis is similar to MS in that demyelination is a primary pathology, but it is monofocal and monophasic in the spinal cord, which is a very nice initial target. And then subsequently, we would look in multiple sclerosis at the spinal cord lesions that are linked to motor disability. So if we look at risk mitigation with our cell therapy product, as I mentioned, it's a localized therapy. We think that significantly reduces the risk of systemic toxicities since they don't leave the CNS. We haven't seen any toxicities in over 200 animals to date. And again, a normal cell product in a native environment, normal support functions, and the fact that we can target multiple indications allow us to leverage our development costs across many indications. So looking at our pipeline, again, the first 3 products are with the Q cells. They're all in the spinal cord, so again, we can leverage a lot of the safety studies from each indication. Down the road, we will be looking at the brain diseases, Parkinson's, Alzheimer's, and stroke. So looking at the competitive landscape in the central nervous system cell therapy space, there really are not many companies. You heard some of these speak in the prior session. Nobody else other than Q is looking at the glial progenitor cell. Neurostem and stem cells and Reneuron are looking at neural stem cells. Geron, as you've heard, has stopped their trials and are getting out of the field. We think there are significant advantages to focusing on the support cells. And market opportunity, goes without saying, is huge if you have an effective therapy. So our near-term milestones, we are just newly public. We're going to file our S1, so the stock will start trading, file our IND with the FDA and start clinical trials, and then obviously human data. So that is our goal, safety as well as potential efficacy markers. To summarize then, we have a very capable and experienced team. We have top leaders in the field that are supporting us. We believe that we have the best-in-class product platform for a variety of products. We talked about all of these factors, so I won't reread them. Very strong patent position, and we are now a public company with a strategic investor, Teva. Thank you.
Okay, so a couple of quick questions. What is the cell? It's a glial progenitor cell. What's its source? Where's it coming from?
We get it from fetal cadaver, similar to the other companies that are working with neural stem cells, and we've developed a method with a monoclonal antibody to isolate the cell type we want, and then we've developed a method to expand that cell.
And what do you how do you move into a phase 1 trial? Do you envision doing dosing? And given the fact that it's an allogeneic cell, when you start to obviously autologous isn't really an option here.
It's not an option. No.
Yeah. So given autologous isn't an option, is the idea that maybe you'll retreat, and how long will the cells be persistent?
We don't know the answer to that question. In native, healthy individuals, these glial progenitors reside throughout the brain and the spinal cord throughout the life of that individual. That's in contrast to a neural stem cell, which really only resides in a certain area in the brain. And they're only weakly immunogenic. There are studies, again, you heard in the last session, where they stopped immunosuppression and the cells survive. There's been studies in Parkinson's patients where they showed that the cells survive 10 to 15 years after stopping immunosuppression. So potentially it could be, a life-changing single treatment, or it could be a yearly treatment. We won't know until we do the studies.
It's very exciting. And these cells, is the mechanism of action, or the theoretical mechanism of action, that these cells are secreting certain cytokines that are then eliciting the surrounding cells to go and remyelinate back to the target?
There are animal studies that suggest that, but they also, the oligodendrocytes that our cells produce, we have shown in those slides that it's actually those cells that are producing the myelin. They're sending their little processes around the axons. So you can have both mechanisms. Certainly, they do produce a lot of growth factors. The astrocytes are known for that. They also take up glutamate, remove toxins. So they really do everything that's needed for neurons to function. I just wanted to make one other comment when you ask on source. We currently source from the fetal cadaver. We do have IP, and we're developing methods to source from induced pluripotent cells for second-generation products down the road.
Sure, because you know what the cell type is, and there's multiple other places where you might be able to get it. So I'm going to ask you to close on something that I find really, really fascinating, and that is the fact that Teva is a strategic investor in Q Therapeutics. So Teva, as we all know, ended up acquiring Cephalon, and Cephalon paid, I believe, about $120 million in cash and a 20% equity stake in Mesoblast. So it seems like Teva is really focusing on the cell therapy space. Can you give us any insight into what Teva is thinking in terms of where they want to be in cell therapy?
I'm afraid I can't, but your summary is obviously what we would like to see, but, I don't